Okay, well, thanks to the conveners for inviting me. I'd like to acknowledge my collaborators who've helped in a variety of ways, some that won't be obvious in this discussion. Also, thanks to colleagues for discussions and others for helping collect the data that I'll be presenting here, and the National Science Foundation and WHOI for funding. So I'm going to discuss data from only 2 depth transects today. I'm going to focus on the Delta C13 decrease that occurred on the LGM to HS1 transition, and I'm going to look at data only from cores deeper than 1,000 meters. The 2 depth transects are from the North Atlantic. Most of the cores are from the eastern flank of the Reykjanes Ridge. We included 1 core that wasn't on the ridge to include data from a deeper core, and our second depth transect is from the Brazil margin. So these are the only time series that I'll be showing you in today's presentation, and these are 5 records from the shallow North Atlantic between 1,450 and 1,650 meters water depth, and I wanted to make sure you knew the feature that I was discussing, which is this sharp decrease on the transition. And that Delta C13 decrease was associated with a Delta O18 decrease, which I won't be discussing today, but is probably related to a decrease in the Delta O18 of seawater and warming, and the relative influence of those 2 processes is dependent on the core's location. So these are all the data that I'll be discussing today. And these are depth transects for LGM Delta C13, Heinrich Stadial Delta C13, and the Delta C13 difference between those 2 time slices. The Brazil margin data are in blue, the subpolar North Atlantic data in red, and the LGM shows features that you're probably all familiar with: the high Delta C13 values in the high latitude North Atlantic, presumably North Atlantic deep water, the low Delta C13 values in the South Atlantic, presumably Southern Ocean water, and in the LGM, on the Brazil margin, there's this third water mass, Antarctic intermediate water, which here is shown by only 1 data point, but it's actually well resolved with shallower cores and also with cadmium calcium. During the Heinrich Stadial, there is no Delta C13 gradient between the North Atlantic and the Brazil margin. David Lund showed this previously. There is a Delta C13 decrease with a depth of about 0.8 per mil. And in this final panel, we see the Delta C13 difference between the 2 time slices, which was largest in the shallow North Atlantic, and its amplitude decreases with depth. The Delta C13 decrease was also largest at similar depths on the Brazil margin, but the amplitude of that Delta C13 decrease was smaller than in the subpolar North Atlantic. And in our Antarctic intermediate water depth core, the Delta C13 actually increases. So we can look at the relationship between the LGM Delta C13 and the Delta C13 difference, and we see a very strong correlation. And the only core that falls off that trend is the core in LGM Antarctic intermediate water. So from this figure, so this points to the North Atlantic as the source of the Delta C13 decrease, as has been suggested in many previous studies, which suggests that perhaps there was a decrease in the Delta C13 of the North Atlantic deepwater end member, or a decrease in the fraction of North Atlantic to depth, or perhaps both of these processes, and as we'll see, perhaps other processes as well. So before I investigate the cause of that Delta C13 decrease, let's look at some water mass mixing diagrams. These are Delta O18 versus Delta C13 in the benthic forams. The Delta O18 reflects conservative properties, the Delta O18 of seawater and temperature, whereas the Delta C13 may be influenced by non-conservative behavior, specifically the remineralization of low Delta C13 organic matter. So in green are the Brazil margin data for the LGM, and we see that the data fall along a single line, with the exception of the Antarctic intermediate water point. They trend from the low Delta C13 values of the deep South Atlantic to higher Delta C13 values. They project slightly below the North Atlantic end member, likely due to the contribution of this glacial Antarctic intermediate water. The North Atlantic data trends from the high Delta C13 of North Atlantic deep water to lower Delta C13 values, with the deep North Atlantic was a North Atlantic northern source, southern source mixture. I've put on the Heinrich stadial data. In orange is the Brazil margin data, and blue are the North Atlantic data. The North Atlantic data trend from relatively high Delta C13 values for HS1 and trend directly towards the lowest Delta C13 values on the Brazil margin. And the deepest North Atlantic, therefore, was a northern source, southern source mixture. During the Heinrich stadial, the data from the Antarctic intermediate water depth core, shown in red, now falls on the trend defined by the deeper Brazil margin data, confirming, in fact, that there was no influence of a third water mass during the Heinrich stadial. What's most puzzling about the Brazil margin data is that they don't trend to the northern source. And so we have to ask ourselves whether North Atlantic deep water was actually advected to the shallow Brazil margin during HS1. Dave Lund suggests that yes, it did, and you should go hear his presentation later in the week to get the details. And so he draws a mixing line between the high Delta C13 northern source and these mid-depth waters lying between 2,500 and 3,000 meters water depth. I'm only showing one of these data points. He has additional data that he'll show in his presentation. And he suggests that the deep South Atlantic was isolated from North Atlantic influence until late in the Heinrich stadial, consistent with earlier studies. So with that background, we're going to investigate the cause of the Delta C13 decrease, and first we're going to assume that Delta C13 behaved conservatively. So our first scenario is that these mixing lines shown by straight lines are the right mixing lines, and they would imply that there was a 1 per mil decrease in the Delta C13 value of the North Atlantic deepwater end member, and that the entire Delta C13 change at our shallow North Atlantic sites, therefore, was due to this decrease, and there was no change in the fraction of North Atlantic deepwater to those sites. Our second scenario, we take the opposite extreme, that there was no change in the Delta C13 of the source value, and that the the HS1 value was the same as the LGM value. To find that data point, we project the North Atlantic mixing line to 1.5 per mil, the LGM value, and these mixing lines imply 60% reduction in the fraction of North Atlantic deepwater to the shallow North Atlantic sites. Okay, so to see whether these scenarios are consistent with the Delta C13 data, we're going to consider 3 hypothetical locations containing 30%, 50%, and 80% North Atlantic deepwater during the LGM. We're going to estimate their Delta C13 value by assuming 2-component mixing between the Brazil margin and the deep Brazil margin and the shallow North Atlantic, and we're just going to pick off the values corresponding to these percent North Atlantic deepwater. To find the Heinrich stadial values, we're going to, for scenario 1, we're going to pick off the percent North Atlantic, the fraction of the Delta C13 corresponding to these fractions of North Atlantic deepwater from the solid mixing lines. And to look at changes due to the second scenario, we're going to pick values off these dotted mixing lines, and they're going to be 60%, 40% of the original water mass fraction at those sites. And when we, and the results of those two scenarios are shown by red and blue dots, and we see that they fall quite close to the data, as does an intermediate scenario where about half of the decrease is due to a change in the source value, so the North Atlantic NADW end member value of 1 per mil, and the rest due to a water mass fraction change. So now we've assumed that Delta C13 is conservative, but we know from water hosing experiments, well, we don't know. Water mass hosing experiments suggest that an AMOC shutdown makes the Atlantic more Pacific-like, and we see this in a lot of water hosing experiments. And the experiment that I'm going to look at today is a hosing experiment on a pre-industrial control run. And the reason I'm looking at this one is because we can look at changes in Delta C13. And these are results from Schmittner and Lund, and they'll be presented later in this week. And what Andreas provided was Hofmeiler diagrams for the site containing, the grid box containing our North Atlantic cores, and we're looking at depth versus time for a variety of properties. The hosing occurred at time 0, lasted for 400 years, during which the AMOC collapsed and remained collapsed even when the hosing was withdrawn. And what we see at this high latitude site is a dramatic cooling in the surface and subsurface. We see freshening, strong freshening down to 1,000 meters as this fresh water is incorporated into the subsurface. We're going to hop over to Delta C13, and we see an increase in Delta C13 at the surface and a decrease in the subsurface, and we see a decrease in phosphate. So even though we're not necessarily advocating a shutdown in the AMOC, this experiment gives insight into what might be causing Delta C13 to decrease at our North Atlantic sites. And as described by Schmittner and Lund, there are two factors. One is when the AMOC is shut down, there's a reduction, almost a cessation, of the input of high Delta C13 water to the subsurface. So this obviously increases decreases the Delta C13 of the subsurface and perhaps contributes to the increase at the surface. And because that water subsurface is being poorly ventilated, there's also an increase in respired carbon. So let's look at the data one more time, and this time I'm actually going to put the regression through the Brazil. This is the regression through the Brazil margin data, and we see that the lines are subparallel. And one simple interpretation of these data is that there was a shutdown, and that the North Atlantic deep water did not reach the upper Brazil margin. The alternative is that North Atlantic deep water did reach the Brazil margin, and to find the northern M member, we project the Brazil margin data this time to a hypothetical North Atlantic deep water M member having a Delta C13 of 1 per mil. So if this was the real N member, then the mixing line between a North Atlantic N member and the southern M member would be something like this green line, and all the North Atlantic data lie below it. And we suggest that if North Atlantic deep water reached the Brazil margin, then this difference from a hypothetical N member would be due to respired carbon. So, and we can make these data consistent with the Delta C13 data as well, because we attribute the offset in the North Atlantic to respired carbon. So to conclude, we hypothesized that an AMOC reduction made the Atlantic more Pacific-like. That's consistent with cadmium, calcium, and radiocarbon data. The Delta C13 at our shallow North Atlantic sites is lower than the true North Atlantic deepwater M member. If this was the case, then southward advection occurred shallower than these North Atlantic sites. I think I'll stop there.
Yeah, thanks, Delia, for this nice and convincing talk. I think we don't need other proxies any longer. Maybe there's one quick question from the audience. If no, then we proceed with Zhengyu Liu from University of Wisconsin, and his talk will be about AMOC evolution and the last deglaciation, forcing mechanisms, thermohaline instability, and implications. That's it.
Thank you very much. Here is the pointer.